Hello, this is Mr. Merrier with Noteworthy Science. Today we're going to be doing the additional higher level component of A1.2 Nucleic Acids. Under the curriculum matrix, this falls under Theme A, Unity and Diversity, and the level of organization of 1 Molecules. Some guiding questions to keep in the back of your mind is, how exactly does the structure of nucleic acids allow it to store information, specifically related to heredity? And the second question we really want to think about is, how does the structure of DNA really inform replication? And while the subtopic is not going to be going into replication per se, specifically, it is going to set the foundation as to how that structure really enables this process to occur. In additional higher level, there are 5 learning outcomes that we're going to be going into. Specifically, we're going to be diving into the structure a lot more in detail, and we're going to be exploring some really key experiments from pretty amazing scientists in the past as to how the structure of DNA was deduced and how we were able to determine that it was in fact nucleic acids and not proteins as the genetic material. Let's begin. So in standard level, we looked a lot about the structures of nucleic acids, right? Specifically, we looked at a lot of the structure of DNA and RNA, and we already started to make links about how the structure of DNA and RNA is related to their function, right? And that's going to be a recurring theme throughout biology. How is the structure related to its function? We're going to be doing that even more today. In order to do that, we have to look a little bit more deeply into the structure of DNA. So we're going to zoom in to the structure of DNA, and now we're going to use numbering systems. So before, when we described the 2-stranded structure of DNA, we said that it was anti-parallel, where one strand was facing up and the other strand was facing down. Now that's somewhat vague in terms of what does that mean, up or down? That's a very relative term. So we have a much better way of identifying the strands, and that is by numbering the carbons. So the way that we number carbons, let's say this was our deoxyribose right here, and ribose is also numbered the same way. Remember that this top part is an oxygen, not a carbon, right? And we said this was a pentose sugar because it was composed of 5 carbons. Those 5 carbons are going to be numbered clockwise, starting from the carbon attached to the nitrogenous base. So this would be carbon 1, 2, 3, 4, 5. Carbon 1 is attached to the nitrogenous base, right? Carbon 3 has the hydroxyl group, and carbon 5 is attached to the phosphate group, right? So this is the way that we're always going to number it. 1, 2, 3, 4, 5. If we now are looking at an entire DNA molecule, let's look at this strand right here. Carbon 1, 2, 3, 4, 5. I'm sure you guys have used this convention in math as well. That little apostrophe we pronounce as prime, right? So this would be 5 prime. This would be 3 prime. So 5, 3, 5, 3. So the way that we can describe the orientation of this strand is we wouldn't say this strand has the phosphate facing up. We would say this strand is oriented in the 5' to 3' direction because it's 5 to 3, 5 to 3. So if we look at the other strand, this would be 3 to 5, 3 to 5. So this strand would be numbered, or this strand would be described as going in the 5' to 3' direction. This strand would be described as going from the 3' to 5' direction. And so everyone would number the carbons in this way, which means that if I said the 5' carbon, everyone knows what I'm talking about here. It's this carbon right here. Are there carbons here as well? Yes. They're irrelevant. We don't care about them. We're only dealing with the deoxyribose. This also means that you can actually deduce the strands orientation. So let's take a look at this example here. Here I have 2 simplified strands of DNA, and the only information I provided to you is this is the 3' end. Deduce what number, 3 or 5, would you give this end, and this end? If this is 3', that's going to be 5'. that means this has to be 3' because it's going in the opposite direction, which means this has to be 5'. So when the DNA actually coils three-dimensionally, it means that one of the strands is going to be moving, that helix is in the 5' to 3' direction, whereas the other helix is in the 3' to 5' direction. You may be wondering, cool story, bro, who cares? Why does any of this matter? 3', 5', are you just being extra fancy for no reason? Well, as I said before, a big theme in biology is trying to connect structure and function. And once we better understand structures, we can kind of understand how things operate a little bit better. And in this case, understanding 3' and 5' really helps explain how enzymes work with DNA a lot better. So enzymes, as we'll discuss in subtopic C1.1, enzymes, they're usually proteins, but they're anything that can increase the rate of a chemical reaction, right, without being used up. We use enzymes for just about everything in metabolism. So the idea is that most enzymes can only bind their substrates in a very particular way. So these enzymes are only going to be able to bind to DNA and read DNA or RNA in a very particular orientation. So let's look at this first example here, DNA replication. We'll look at this process in D1.1 more specifically, but when we replicate DNA, what we're doing is we're taking a strand, separating it, and then each of those strands, remember, are used as a template, as we talked about earlier. That was one of the big benefits of complementary base pairing. The fact that if I have one strand only, I can deduce its complementary partner, no problem. Why does the 3', 5' matter? Well, the major enzyme that's going to be linking new nucleotides, DNA polymerase III, this operates only in one very particular way. Let's say that this is 2 DNA strands. This is 5 to 3, 3 to 5, right? So DNA polymerase III, and actually many of the other enzymes involved in DNA replication, can only attach a new nucleotide in a very particular way. They can only bind this to the 3' end. They're never going to attach this nucleotide to a phosphate or the 5' end. Remember when we looked at condensation reactions, we said that this had an OH right here. This had an OH right here, right? So what's going on here is that this and this are ultimately going to come out in order to link it together. Now, you might be like, couldn't you also just do the same thing with the OH here and there? In reality, yes, but these proteins, like I said, have a very particular shape and a very particular active site, and the way in which they've evolved is they can only really operate in this fashion. So this protein can only link a new nucleotide to the backbone at the 3' end. I've tried to illustrate this just a little bit right now. So in purple, I have my parent DNA, the original DNA. Here's the 5' end, here's the 3' end. This thing is being split open, right? And this enzyme effectively can attach to the 3' end, 3' end, 3' end, 3' end. Thus, you can describe the process as occurring 5' to 3'. Because I'm consistently adding to the 3' end, then you can describe this action as happening 5 to 3, 5 to 3, 5 to 3. So it's attaching or binding to the 3' end, but you can describe its activity as it is replicating DNA in the 5' to 3' direction. So if you look at this process, that means that this strand, this enzyme is cooking. It's going to replicate as quickly as this separates, no problem. As this thing opens up, it's going to continually replicate. But this strand, you can't do that. It can't add to the 5' end, so it has to almost open up, replicate, open up, replicate, open up, replicate. So this end is going to be replicating much slower than this end. Again, we'll explore all of this concept in D1.1. But as a preview, this replication is very fast, or something that we can call continuous replication, whereas this replication is kind of disjointed, something that we call discontinuous replication. And so you have to replicate in pieces. The reason for this is because these enzymes can only bind in a very particular way. So let's look at transcription. Here in purple, again, I have the DNA strand. Now I'm going to use the enzyme RNA polymerase. That's going to separate the DNA, and I'm going to use one of these strands as a template strand. Again, this process we'll be looking at more deeply in D1.2, or protein synthesis. So this means that this new mRNA is going to link a new RNA nucleotide to the 3' end, 3' end, 3' end, which means that the process is occurring in a 5' to 3' direction. So both DNA replication and transcription both occur in the 5' to 3' direction because the enzymes are linking a new nucleotide to the 3' end of the previous nucleotide. The last process here is translation. Translation is where the mRNA transcript is going to be delivered to the ribosome. The ribosome is then going to read the mRNA, and as it's going codon by codon by codon, amino acids are being built together, and you're synthesizing a polypeptide. Now, this process also occurs in the 5' to 3' direction because this is the order that a ribosome is moving. Let's say I have something called a polysome. In red, I'll demonstrate, here's my mRNA sequence. And then attached to this mRNA are a bunch of ribosomes, where you have one mRNA transcript and they want to make a lot of that protein, so a bunch of ribosomes are all going to bind. Look at this diagram and deduce which end of the mRNA is 5', which end of the mRNA is 3'. So we know that because this is the shortest polypeptide, this translation process must have just started to occur, whereas this one's nearly done. So even though I didn't show direction of movement, you can infer direction of movement based off of how long the polypeptide chain is. And if you remember that the ribosome is effectively moving through or reading mRNA in the 5' to 3' direction, you'll be able to deduce the relative orientation of these molecules. Right? Draw a DNA molecule comprised of 4 nucleotides and label the directionality of both strands. Your DNA molecule should look something like this. If I were to label the directionality, I'd have to say, well, is a strand on the left going 3 to 5 or 5 to 3? Well, let me find the 5' carbon. Here is the 5' carbon. Here is the 3' carbon. So this is going 5 to 3, 5 to 3. So I could say this strand is going 5' to 3'. On this strand, I can see that this right here, this is my 5' carbon, this is my 3' carbon. So that would be 3 to 5, 3 to 5. So this strand is going 3' to 5'. So on a test or on a whatever, if you see a diagram, you should be able to deduce where's the 3' end, where's the 5' end. So for instance, if I said, where would a nucleotide bind on the left-hand strand? It would have to bind here. Here's the 3' carbon. That's where a hydroxyl group is. And then a new nucleotide here's a new nucleotide. That means that if these were actually to bind, this is not going to bind to this part. The enzyme can only attach to the 3' end, so a condensation reaction would happen here, and that's where this new one would bind. It would bind to the 3' carbon. Something that we've looked at in standard level is that of complementary base pairing. And you might be like, bro, again, we just talked about this a lot. And it's not too much more actually in HL. I'd say the key difference is that while in standard level I was using terms like purines and pyrimidines, HL students are required to understand the difference between a purine and a pyrimidine. So a purine are the 2-ring structured nitrogenous bases. So that would be adenine and guanine. The pyrimidines are the single-ring structured nitrogenous bases, and that would be thymine, cytosine. Uracil would also be a pyrimidine. So the idea is that DNA binds in a very particular way. It binds, we already know, complementarily, but beyond that, it also is a purine bound to a pyrimidine. This is really, really key at ensuring the stability of a DNA structure, something that we've explored also in standard level. But the idea is that if you've had 2 purines together, then the structure at that point would be really large. If you had 2 pyrimidines together, the structure at that point would be really narrow. The structure of DNA would become quite unstable. The fact that a purine and a pyrimidine always bind ensures that the diameter of a DNA molecule is relatively constant, which is about 2 nanometers in diameter. So even if you look through a DNA polynucleotide that's millions of nucleotides long, it's a very consistent, stable structure. And a big reason for this are these stable hydrogen bonds in the middle, holding a purine to a pyrimidine together. And this is a main contributor to the stability of that DNA double helix. Looking into more the structure of DNA, but specifically within eukaryotes. Prokaryotes and eukaryotes both use DNA as their genetic material. The way in which DNA is packaged is not the same between eukaryotes and prokaryotes. We mentioned already that DNA is a fantastic molecule for storing information because, well, for a lot of reasons, right? We said it was variable in length. We said that it has a very small diameter, which means that there's a lot of information that can be packed. And we said that DNA is very, very, very compressible. If you took all the DNA, recall, out of your cell and stacked it end to end, it's over 2 meters long. That's an enormous length to fit in a 10 micrometer diameter nucleus. That's crazy. So a way that we can do this is the way in which DNA is packaged. So the genetic material in your nucleus is not just composed of DNA, it's also composed of proteins. And these proteins are called histones. I'm going to show you a simplified diagram here, and then we'll explore this more at the 3-dimensional level. So here I've shown with these 2 blue stripes here, this is a simplified version of DNA, okay? And I'm showing it, notice that it's being coiled around this 8 subunit protein complex called a histone. So there are 1, 2, 3, 4, 5, 6, 7, 8 histones, or something referred to as an octamer, oct for 8, structure where DNA is wrapped around. There's a 9th histone called the H1 histone that sits on the outer end, and that kind of holds it all together. So the way that you would describe this is DNA is looped around an octamer of histones with a final H1 histone kind of locking it together. Now, if I can describe this as a histone and this as DNA, I would describe this whole unit as a nucleosome. So a nucleosome refers to both the DNA and the histone. So if I said the nucleosome, I'm referring to DNA being wrapped around this octamer of histones. That's what I'm referring to. The DNA that's literally linking one nucleosome to another nucleosome is unsurprisingly called linker DNA. So this model of DNA structure is sometimes referred to as the beads on a string model because it's a bunch of these in end to end to end to end to end. So it's kind of remarkable how compressed this can become, where you take, here's one nucleosome, here's a bunch of nucleosomes together, they become packed in this structure that's high in DNA and protein called chromatin. that coils on itself even more, which further coils on itself, and that's the structure that you can maybe see when we're looking into microscopes, chromosomes. So a chromosome really is just one large DNA molecule that has been hyper-condensed. We're going to look at this when we look at mitosis and meiosis, but the only time that you're going to see DNA arranged in this way is when it's about to divide. Normally, it's going to be in a looser, uncondensed structure, because how am I ever going to do gene transcription in a hyper condensed state, right? It's almost like in a computer, good luck working on a file that's as a RAR or a zip. You have to first uncompress it to then use it. The same thing is true for DNA. We're only going to hyper compress it in this way when we're about to divide a cell. Normally it would be slightly uncompressed, but still these histones act as organizing structures. So the DNA wraps around it. Now these histones are not just important for structure. In another topic that we'll look at in this course, gene expression, they have a huge role in actually, well, expressing your genes, because something that's a really fascinating area of biology, something called epigenetics, it's how does the environment alter gene expression, right? The idea that depending on what you eat, where you live, what you do, that environment around you can actually modify your gene expression. And they do that by modifying your histones. Your histones can actually take these tags that either cause some sections of your DNA to be expressed more or less. But histones are not just useful for organizing, they're also really key in gene expression. It's a little lame to try to understand nucleosome structure with a 2D dimensional representation like this. So there's a few links here that I'd like us to explore. Let's go to this top link right now. In gray is a nucleosome, and notice that you have the DNA wrapped around it. This little end coming off of it is just a histone tail, and that's where I was telling you before, that's what actually can be modified by the environment. What's not being shown, of course, is multiple nucleosomes and the linker DNA. So if I were to show another nucleosome, it would be right here, and then the linker DNA, if you find the end, here's an end of DNA right here, this would link to the other nucleosome right here, and there would be an H1 histone locking that in place. So when we look at this guide here is actually quite good because it can run you through the actual structure all the way in. If you click on that, move DNA away just so you can see the histone now, and you can see 8 different sections. So the 8 histones actually are 4 pairs. So it's almost like 4 dimers, dimer for 2. And that's what makes up the octamer, or this zone right here. So this is the actual histone right here. This is all protein. And if you really zoom into one of the histones, something that we'll look at when we do proteins, this will make a lot more sense when we study proteins specifically. But if I were to zoom into just a single one of these histones, then this is one histone. This is one polypeptide. And that's the way that polypeptide will actually loop in this 3-dimensional structure called an alpha helix. And that's what makes up that one protein. So there's 8 of these proteins that make up that core. And that's what DNA is looped around. The question is, what charge would you say DNA as a molecule would have overall? And once you know that, what kind of properties must histones have to be able to associate with DNA tightly? Let's zoom into here. Phosphate groups actually carry a negative charge. When you look at the entire molecule together, you'll notice that the phosphate backbone on the outside is actually negatively charged. So if the outside of DNA is negatively charged, then we can deduce that some areas of the histones must be positively charged. The peripheral, meaning the outside positions of these histones, actually carry a slight positive charge. And that allows them to bind or link onto DNA quite strongly, and that's what holds them in place. It doesn't mean the entire histone is positively charged, but those areas that are strongly associated with DNA are. Any of the parts that would be touching DNA, so to speak, that part of the protein will be positively charged, and that's what allowing it to hold on well. It doesn't mean the entire protein is positively charged, but some areas will be. And this, I think, will make more sense when we look into B1.2, when we look at the structure of proteins. And you can see that some of their amino acids might allow them to be positive or negatively charged on one area, but some of their amino acids may be uncharged. So it doesn't mean that the entire protein itself is one charge. Sometimes different parts have different properties. This third link right here is also really useful because it allows to examine nucleosomes in different styles. So this style right here, you'll notice here's the DNA in purple, as you can clearly see with the helix. Here inside are all the alpha helices that make up the proteins. But if you want to change the style, and I want to, let's say, do a backbone projection, now this is basically, I only want to see the simplified structure. So here you can clearly see the double helix. If you wanted to see a more, what's called a space fill projection, this takes, again, into account all of those electron shells, so it looks slightly bulkier. I can label by chain, by properties, or even by amino acids if I wanted to. And you can really start to see, for instance, by chain, here's one of the DNA chains, here's the other DNA chain. So these kind of projections are really, really useful at trying to better understand how something actually works. They're labeling the proteins by different amino acids. Each of those amino acids are given a different color. And this really allows you to really just examine the structure better. So I would always, at the end of my lessons, I'll always link three-dimensional structures for you to explore further, because I can only do so much in a 2D projection, but when you really start to look at this from a three-dimensional point of view, I think it really helps better inform kind of what's going on here. I think this part is often neglected in science, and that is, how did we get to where we understand this today? What were the experiments that led to these discoveries? This experiment, the first one that we're going to talk about, is honestly, in my opinion, such a good example of creativity within science. I think oftentimes people that enter STEM or enter the sciences are accused, or maybe the stereotype is that these are the non-creatives. These are like the robots that only care about logic and whatever, and there's no creativity. Creativity is for the arts. I disagree strongly with that notion. I think science requires a lot of creativity because it requires ingenuity as to experimental design, for instance. And hopefully after today, you're going to see what I mean. Let's rewind back the clock here, and let's go back to the 1800s. And in the 1800s, we were at least confident that chromosomes played a role in heredity, meaning that they were used to transmit information from parent to offspring. We knew that. And we also knew that chromosomes, as we just discussed earlier, are composed of both DNA and proteins. Now, what was not known, or at least confirmed, is, well, which is it? Is it proteins that are responsible for genetic information, or is DNA responsible for genetic information? Now, obviously, we have the benefit of hindsight, and we're obviously like, of course it's DNA. But if you're then and you don't know, how do you know it's DNA and not proteins? How do you come to that conclusion? And in fact, it might surprise you to know that proteins were actually the front runner. They were the molecule that most scientists were betting on, so to speak, as to being the genetic material. Because if you compare proteins and DNA, proteins are significantly more variable, right? There's 20 amino acids versus 4 nucleotides in DNA. They're much more complex structures than DNA can be. So a lot of scientists were hypothesizing that it was proteins that was genetic material, not DNA. So how did we actually figure out that, in fact, it's not proteins, but DNA that is the genetic material? And that's this first experiment that we're going to talk about, called the Hershey-Chase experiment, named after the two major investigators in this experiment. So this experiment, I think, for me anyways, is probably one of the best examples of creativity, of just like how did they come up with this is kind of amazing. So to understand how this experiment works, we're going to do a slight tangent and understand how viruses infect. So we're going to be looking at this specific virus in the virus subtopic, but this is what's called a bacteriophage. This guy, this is a bacteriophage. So a bacteriophage is a virus, or the T2 specifically, infects only E. coli as a bacterium. So there's likely billions, trillions of this virus in you right now, but it's inconsequential because they do not target your cells. They only target bacterial cells. And they've been around for billions of years. They're very, very efficient. And this was known even at the time. They were aware that this virus infected E. coli by binding onto it and injecting genetic material. They didn't know what it was injecting. Was it injecting proteins or was it injecting DNA? We don't know. But we do know that once it injected its genetic material, that genetic material basically was then used by the host without really its knowledge or will, and it basically just becomes a virus-making machine. And then it makes more and more and more and more viruses, and to the point where it can actually split open and start the cycle anew. This is what was known. The main question was, is this T2 virus using DNA or proteins? Now, why use viruses? Well, viruses are simple in the sense that they're only made up of DNA and proteins. That's it. So the structure of a bacteriophage is a protein shell called a capsid and the DNA inside of it. That's it. So we want to use this as a model organism to try to determine, are the viruses using DNA to infect, or are they using proteins to infect? Well, how can I possibly realize this? And this is going to be a great example of another recurring theme that we're going to talk about in science. Knowledge in science is often limited by technology. As technology improves, our ability to observe improves, and thus our understanding can improve. So many different discoveries in science really are all linked to a specific piece of technology that arose, right? Consider everything that we've learned about cells after a microscope was invented. Consider all the things we've learned about space when we use the Hubble telescope or the James Webb telescope. Consider what we've learned about particle physics through the Hadron Collider, right? Anytime that there's a breakthrough in technology, there's usually a breakthrough in scientific understanding. And in this case, it's about radioisotopes. So we've discussed isotopes previously. Now a radioisotope is a special kind of isotope where one of its isotopic configurations is unstable or radioactive. And that means that it will undergo what's called radioactive decay, and where it will emit effectively radiation or energy. We don't have to go into that too much. Those of you studying physics will be going into that more. But the idea is that as it decays, that radiation is being emitted. Now, we can detect that piece of radiation using certain devices, one famously named after its creator, Geiger counter. That's the thing that clicks. You guys know what I'm talking about? When you sweep it over and it goes like, and it clicks like that. This will detect radiation levels. So we can label certain materials with a radioactive isotope and then detect where that material is going by detecting the radiation that it's emitting. The reason why this is probably the first nature of science, or NOS, learning outcome, that we've discussed so far. And when we say the nature of science, what we really are talking about is any learning outcome that has to do with basically how did we get here? What piece of technology was used? What kind of hypotheses were used? What kind of thinking was used? So the idea here is that the nature of science learning outcome that they're referring to is that this whole experiment would not have been possible without radioactive labeling becoming a piece of technology that was available. Now, I believe radioactive labeling was discovered in the early 1900s, but it wasn't really readily used until World War II. And that's, probably largely associated with the Manhattan Project, and our understanding of radioactivity started to increase dramatically. And then post World War II, this technology became much more widely available for researchers. Now, Hershey and Chase had the technology of we can use radioisotopes and we can detect where they go. So we're going to use two different radioisotopes. We're going to use radioactive phosphorus, or P32, and we're going to use radioactive sulfur, or S35. I'm going to label DNA with phosphorus, that's radioactive, and I'm going to label proteins with sulfur, that is radioactive. Now, why use phosphorus for DNA and why use sulfur for proteins? So I want to be able to label where is this DNA going without labeling other things. Well, phosphorus is not found in proteins, but it is found in DNA, in the phosphate group. But sulfur is found in proteins, it is not found in DNA. So I can label the DNA with radioactive phosphorus, and I can label the protein with radioactive sulfur. So this experiment was done in two major phases. The first phase, I'm not going to label the protein, but I am going to label the DNA with radioactive P32. And then in another experiment, I'm going to radioactively label the sulfur, or the protein, with S35. And I'm not going to label the DNA. So I'm only including radioactive phosphorus in here. I'm not labeling the protein. This, I'm only labeling the protein, not the DNA. So here's what's going to happen. I'm now going to allow this virus to infect E. coli. I know that it's going to inject genetic material. I don't know which. Then I'm going to basically put this into a blender, shake it up a lot, and that's going to dislodge the viruses from the bacteria. Hopefully now whatever genetic material is in here now. And then I'm going to use what's called centrifugation. Basically it's fancy for if I take something that's a mixture and spin it really fast, what happens? So centrifugation separates by density. So you start to get a density gradient. The most dense goes to the bottom, the least dense go to the top. Both of these tubes get spun very fast. What you get is you get this denser part called the pellet and this liquidy part called the supernatant. In the pellet, what you're going to find are what's the dense part of this? All the bacteria. So all the bacteria are going to be found in the pellet, and the much lighter supernatant is where you're going to find the viruses. If you hypothesize that proteins are the genetic material, where should the radioactive phosphorus be here, and where should the radioactive sulfur be? Where should radioactive phosphorus be here? In the supernatant. DNA was not being injected into the bacteria, so it remained in the viruses. If proteins were the genetic material, in the pellet, because proteins were being injected into the bacteria. Now, let's do another hypothesis. If I hypothesize that DNA was the genetic material, same question. Where would you expect radioactive phosphorus to be? Where would you expect radioactive sulfur to be? If I'm predicting that DNA is the genetic material, then that's implying that the bacteriophages injected DNA into the bacteria, meaning that radioactive phosphorus should be found in the pellet, and radioactive sulfur should be found in the supernatant. They found that radioactive sulfur was found in the supernatant, not the pellet, suggesting that the bacteriophages were not using proteins to infect E. coli. So what then would you find here? They found the radioactive phosphorus was found in the pellet because it was contained within bacteria. Some was still found in the supernatant because it's possible that some DNA remained in bacteriophages and maybe not all of the bacteriophages successfully infected, but the vast majority of the radioactive phosphorus was found in the pellet. So what this experiment conclusively was able to show is that DNA, not proteins, is the genetic material. So again, for me, this experiment is fascinating in how do you come up with this? It's actually quite complex. You're using a mechanism, a real-life mechanism of viruses infecting bacteria, and you're using this new technology of radioisotopes, putting all of that together to answer a really important question. We now know that DNA, not proteins, is a genetic material. Let's go back in time and let's look at, okay, did we always know the structure of DNA? That experiment was saying, was it DNA or proteins that's actually used? This experiment is now going to be looking at the structure of DNA. So Levine, as a scientist, was like kind of goated in how much he's contributed to our understanding of DNA. It was he who coined the term nucleotide. He discovered, and his associates discovered, deoxyribose as a sugar in the first place. He's the one that discovered phosphate sugar backbone. So he got a lot of things right. This guy had a lot of research done and contributed a lot to our understanding, but like everyone else, at some point in their life, he was wrong as well. And he proposed something called the tetranucleotide hypothesis, which there was this repeating order of nitrogenous bases. So he would have predicted that the nucleotide ratio of adenine, thymine, guanine, cytosine would be 1 to 1 to 1 to 1. Well, the seminal experiment that falsified this idea is Chargaff's experiment. So Chargaff used methodology that we are going to be doing later when we study plants and photosynthesis, C1.3, when we're going to be doing a technique called chromatography. Chromatography is a technique, chroma means color, right? It really, actually, I think the word literally means writing with color, but chromatography is a separation technique where you take a mixture, you allow a solvent to move through it by a capillary action, and because the solubility of the different stuff within the mixture is different, it separates the components of that mixture. So on a paper, you're going to see as the solvent moves up, it separates the components of that mixture. Chargaff decided, okay, if I want to try to see is the tetranucleotide hypothesis supported or falsified, I'm going to take a sample of DNA from an organism, and I'm going to separate that DNA out into its nucleotide components. And I want to see what is the proportion of adenine, thymine, cytosine, and guanine, and is it in fact 1 to 1 to 1 to 1, or is it not? How this technique works is you would place your sample of DNA here, you would use a solvent, that solvent then travels up this paper through capillary action, and the different nucleotides would be separated. You would then actually use something called UV spectrophotometry. Here are the results. This experiment examined the proportions of adenine, thymine, guanine, and cytosine across a number of different organisms. Humans, octopus, yeast, maize, wheat, E. coli, and this isn't even all of them. He did more than this. What pattern do you see here? Almost all of these are extremely similar in number or proportion, and these are extremely similar. If the tetranucleotide hypothesis is in fact true, then this should be relatively the same across the board. But it's not. And what's more, add purines to pyrimidine, purine to pyrimidine. This experiment is a seminal experiment that not only falsified the tetranucleotide hypothesis, but also put forward the idea of complementary base pairing. In fact, that idea is actually named after Chargaff, as called the Chargaff's rule. I'm doing a DNA sample on an organism, and that DNA sample was found to contain 15% adenine. Deduce the relative percentages of thymine, cytosine, and guanine. We know that adenine and thymine form complementary base pairs with each other. So if there's 15% adenine, there must be 15% thymine. This combined is 30%. That means that what you have left is 70%. These are also going to be in 50-50 ratios, which means that 70 divided by 2 would be 35%, 35%. Good question was asked is this Chargaff's rule also applicable to RNA, not single-stranded RNA? Because there is no complementary strand across from it, knowing the percentage of one of the base pairs does not actually provide you any information as to the rest of the molecule. Now, as a very last point here, this is a great example to mention how knowledge is obtained in science. And something that will kind of trigger all scientists the world over is the use of proof. We don't prove things in science. We can support ideas or we can falsify ideas. Chargaff's experiment is a great example of falsification. And all of that is based off the fact that scientific knowledge is effectively built using inductive reasoning. So what inductive reasoning means is I make an observation, I detect a pattern based off my observations, I can then form a generalization, and then I can make conclusions about something that I have not seen yet based off of this generalization. Inductive reasoning does not allow you to ever be 100% sure. And that may be a little unsatisfying, but that's kind of what science is. The major drawback to inductive reasoning is that it cannot give certainty. What it's doing is that it's giving you a generalization that is in fact tentative. It's almost like, as far as we know, this generalization holds true. And basically, unless otherwise falsified, this is the explanation, this is the thing that we understand. And that's how inductive reasoning works. So that's why even though we can't prove things in science, we can falsify them with counterexamples. All scientists should be open to being wrong and open to alter their ideas. And this is obviously much harder than it sounds because scientists are people too, and if the thing being falsified is my idea, well, I'm going to be personally invested in that not happening because that's my reputation. So it takes a long time sometimes for scientific ideas to change. When it finally does, that's called a paradigm shift. When everyone is like, you know what, this idea that we had was wrong, we're going to change our understanding. This happened multiple times throughout history with respect to science. Something like the atomic model, something about the heliocentric view of the solar system, right? It used to be geocentric, where the Earth was the center, where it was like, nope, and that took a very long time to finally come across. So the idea is that once you have these ideas that are ingrained, it becomes very difficult, as you can imagine, to try to move away from that. But science as an endeavor should always be open to the idea of being incorrect, as long as you can provide evidence. If you can provide evidence that is, this is actually incorrect, then we have to revisit. And that's why science is an evidence-based endeavor and why it relies on falsification, not proof. And that's, I think, is a good example here.
Let's go over the additional higher level learning outcomes for subtopic A1.2 Nucleic Acids. We started off by exploring the structure of DNA and RNA more in detail, and specifically, we're no longer satisfied by just saying that the strands are upwards or downwards, but now we're specifying the directionality using carbon numbers, or 3' and 5'. And this isn't just a fancy new way of numbering it, it actually has profound significance for a lot of key processes in biology, such as DNA replication, transcription, and translation. We then further explored the purine-pyrimidine bonding and the fact that because of complementary base pairing and because it's always a double ring and a single ring bonding together, it makes the DNA helix stable, having a consistent diameter, regardless of the base sequence. We then explored how DNA is actually packaged within eukaryotes in a structure called a nucleosome, where DNA is actually wrapped around a core of, or an octamer of histone proteins, and we further examined that using 3D modeling. Finally, we looked into a very key experiment that was able to provide strong evidence that it was DNA and not proteins that is the genetic material, and that being the Hershey-Chase experiments where they utilized bacteriophages and E. coli. And finally, we explored how the prevailing notion of a tetranucleotide hypothesis, that being how all the DNA base pairs would be in a repeating 1-to-1-to-1 ratio, was ultimately falsified using Chargaff's experiments using chromatography. And in this way, we were able to further explore the true nature of how knowledge is built within science, i.e. inductive reasoning, and how things aren't proved, but rather they can be supported or falsified. If you wish to learn more, which I always encourage you do, then this page is a good place to start. These are the textbooks that I've consulted and the relevant page numbers that I use to actually build the different content of this lesson. These simulators and interactives are really recommended for being able to look into the structure of base pairing and DNA structure, and specifically, if learning about seminal experiments and how we built our understanding of DNA is of interest to you, then this DNA timeline is a really nice curated look through time as to how knowledge was built over time by a lot of key scientists and experiments. 3D modeling, as we've looked at already, especially at the higher level, is so invaluable at being able to truly understand the structure of different molecules, and I really would recommend that you explore these. Finally, these articles are a really good look into some of the aspects that we've discussed already. Lastly, I would always encourage you not to learn for the sake of a test, but learn for the sake of enjoyment, learn for the sake of learning.